Hi, everybody. So up to this point, we've been talking about how molecules are categorized. We've got inorganic molecules that lack either carbon or hydrogen or both. We have organic molecules that have both carbon and hydrogen, like carbohydrates, lipids, proteins, nucleic acids. We've talked about the types of carbohydrates, monosaccharides, disaccharides, and polysaccharides. We will be moving on to talk about lipids and proteins in this chapter. So we'll continue to categorize those molecules, but we're going to take a little detour today and talk about how monosaccharides and disaccharides can interact with each other, or polysaccharides for that matter. So I want you to look at this molecule right here. You should be able to recognize that. That is a monosaccharide. And here's another monosaccharide. We can tell it's not a disaccharide because they're not connected to each other, right? We've got two separate molecules. Each one is a single ring. So we have two monosaccharides. And then this molecule down here, right, is two rings, but they're connected to each other now. So that's a disaccharide. Now, if you have a plant that's been busy making glucose, a monosaccharide, and has tons of them, they can hook them together into a disaccharide. And how do they do that? Well, the reaction that they do is called dehydration synthesis. Let's take a look. Now, if you look at this molecule, if you count all the C's and the H's and the O's, you will find it has 6 carbons, it has 12 hydrogens, and 6 oxygens, C6H12O6. And we've got another one here. If you look at this one, we have 12 carbons, we have 22 hydrogens, and we have 11 oxygens. So again, to recap, we've got C6H12O6 here, we've got C6H12O6 here, and if we count all of the C's, H's, and O's, we find there's 12 H's, 12 carbons, 22 hydrogens, and 11 oxygens. And there's something funny about that. Stop and look at that for a moment. Do the number of carbons make sense? If you take this ring and you hook them together, does it make sense here? Well, yes, you've got 6 carbons on this side and 6 carbons on that side. So the fact that there's 12 carbons there at the end, that makes sense, right? But look at the hydrogens. We've got 12 hydrogens in this one and 12 hydrogens in this one. That means we should have how many hydrogens left? Well, you can't have them appear out of nothing and disappear into nothing. So there should be 24 hydrogens. But how many are there? There's only 22. We lost 2 hydrogens. 2 hydrogens have gone somewhere else. So when these two rings interact, and that happens, there's a couple of hydrogens that are going to be in another molecule that's just not shown here. But it will exist because you can't make hydrogens disappear into nothing. So when these combine, and this 12 hydrogens and this 12 hydrogens are added together, and you only end up with 22 in this molecule, 2 must go somewhere else. What about the oxygens? We have the same problem with the oxygens. Look, there's 6 oxygens in this molecule and 6 oxygens in this one. So there should be 12 oxygens here, but there's only 11. So along with those 2 hydrogens, an oxygen went somewhere else. Let's take a look at how that worked. Right there, that little OH is taken off, and that H is taken off. When you take that OH and that H off, then you can combine the 2 monosaccharides into a disaccharide because this carbon and this carbon will be attached by the single oxygen in between. Well, what did we take off? What are 2 H's and an O? We are removing water. So we take water out in order to bring these 2 together. So we dehydrate. That means to remove water. We dehydrate to synthesize something larger. So in other words, you can think of it like this. We are getting water the heck out of the way so that these 2 monosaccharides can hook together or perform synthesis, building up a larger molecule. The removal of water, the word for that is dehydration, and the word for making larger things is synthesis, and that's what we're doing. We're taking small things and combining them into a big one, so that's synthesis. And we're doing it by getting water the heck out of the way, so that's dehydration. you can see the full reaction. Here you've got a monosaccharide and a monosaccharide. And move myself out of the way a little bit there. And they combine to a disaccharide and you end up with water. Now, what about if we wanted to do the reverse? What if we ate sugar, sucrose, and we wanted to digest it into 2 monosaccharides? What would we have to add to our disaccharide to digest it into 2 monosaccharides? Water. If we take this H and put it on there, and this OH and put it on there, if we shove water in between, we can bust them apart. And so it's just the opposite reaction. And again, we have water, but this time instead of dehydration, we're going to have the word hydro, the prefix hydro. And here comes a new word part for you that you should know, and that is LYS. Anytime you see LYS, you should be thinking breakdown. So this is the breaking down of something by shoving water in between. Hydrolysis. Hydro is adding water and lysis is breakdown. So in other words, if we take a look at our disaccharide, it is C12H22O11, and we add in 2 H's and an O, we're going to have enough to have a C6H12O6 and another C6H12O6. We're going to take this hydrogen and put it there, and we're going to take this OH and put it there, and we're going to break that bond right there, and we will have broken down our disaccharide into 2 monosaccharides. This can be done with making polysaccharides. We could attach another monosaccharide here by taking away a water and making a bond. We could keep doing it. Or we can break up a big starch molecule by shoving water in between each of the monosaccharides, right? We can take a big polysaccharide and shove water in to break each of them apart by hydrolysis, or we can take a whole bunch of monosaccharides and take away water from each of them as we cook them together into long chains. And that is dehydration synthesis and hydrolysis, and how cells can convert monosaccharides to disaccharides to polysaccharides by dehydration synthesis, or polysaccharides to disaccharides to monosaccharides by hydrolysis.